Welcome back, everybody. This is the RV Falkor during the third expedition to the Great Barrier Reef and Coral Sea, called Northern Depths of the Great Barrier Reef. Our dive here is in the Rodda Canyon up off Cape York. We had some technical problems earlier, which we had to resolve, and now we're back on the side of this canyon, the Rodda Canyon, very large canyon that's draining the edge of the Great Barrier Reef. So-called Rodda Canyon because this canyon head sits between Rodda and Wilson Reef, just opposite Princess Charlotte Bay. Myself, I'm Robin Beaman from James Cook University, based in Cairns. Sitting next to me is Joan, one of our students who is joining us on board. And we're just getting ourselves settled, actually just having a look at this this sort of orange tube to try and figure out what it is. So we welcome you back for anyone who's trying to follow the live feeds from ashore. But very shortly, as soon as the ship is repositioned, we're going to start manoeuvring again upslope. We've got still a fair distance to go. We've got about a good 300 metres vertical rise to go before we end the dive. So just having a look at these orange these long tube-like tubes on the seafloor. Trying to figure out what they are. There's plenty of little these little sea pigs, and also polychaetes. Looks hollow to me. What do you think? Polychaete tube? Yeah, I think we're thinking a polychaete tube, right? An infaunal polychaete tube. There's lots of them lying around, because we know, some of them are calcareous. They can actually cement their own tube. And then glue it to the sides of rocks, and others they just use the surrounding sediment to create a tube. They like glue together all little sand particles to make a tube. Thanks, J-Rod. We can keep going. Yeah, we don't have to keep that. All right, we're going to head up slope. It's quite steep here, clearly very muddy. We're on the lookout for any exposed hard rock outcrops. We're seeing the occasional soft coral, bamboo corals, and we have been sampling as we've been going along, mainly for anthozoans, so black corals and the like. Okay, this is interesting. Soft coral coming into view. But we're going to get a bit of a move along. Still got a fair way to go. And we're heading up the steep sidewalls of this Rodda Canyon. We expect to see it plateau out a ledge around about 1,600 metres, so not far further up from where we are now. And then it'll flatten, and then we expect another fairly steep rise up to the crest of this canyon. And the crest separates this canyon from the adjoining canyon. And we're keeping a close watch with our principal investigators ashore, like Jeremy Horowitz, who's in Townsville watching, and we're getting messages from him as we find animals like colonies like black corals. We're wanting to take samples of those, bring them back to the surface and preserve them. We've got two techniques, one of which is to use ethanol, a sort of standard way of preserving them. Another sampling we're doing is for a big genome mapping project where we're actually preserving the animals in ethanol, in liquid nitrogen. So the sidewalls of the canyon clearly very muddy. We're seeing these little gullies, quite distinct ridges coming into view. Since we went down with that technical problem, we're now up and running again, and we're seeing the odd halosaur, a few other eels, some soft corals. But we're going to get a bit of a move along just so that we can get through to the end of this transect, which is about 1 kilometre long. Yeah, so Joan, you spend most of your research up in the shallow coral reefs, don't you?
Yes, we did most of the transect either by walking in the tidal zone, or we go snorkeling or diving normally above 18 meters.
Yeah, okay.
And JCU is usually a little bit more strict than that, and they would prefer us to stay shallower than 10 meters.
When you're scuba diving?
Scuba plus snorkel, yes.
Yeah, okay. Yeah, pretty shallow. Yeah, so we're at 1,657 metres.
Yeah.
Pretty amazing, isn't it? It does feel like you're actually in the water because the imagery is so clear. Ooh, what have we got here? Is this one of these tripod fish? Can we just have a quick look? We're just dropping the SuBastian down a little bit lower here.
Wow.
Yeah, so we're getting a really a downward look at a tripod fish, and they have this very strange, I think modified pectoral fins, where you can see it's almost they're branched, and they're sitting on, they sit up off the seafloor, obviously looking for food coming past. They're probably an ambush predator. I'm sure there's fish experts at ashore who can tell us more about these tripod fish, but I would think that these modified fins, modified spines, can actually, they'd be sensitive to any animals that drift past, and then they can go and grab them. And if you have a look around the sides of the tripod fish, you see all these little sea pigs. They're quite small, probably about no bigger than your thumb. There's lots of them, clearly making good use of all the soft sediment that's here. Yeah, look at all the, I guess, rays. With the rays are the softer parts of a fish's fins. Spines, of course, are the more bony part. So I'm curious as to, the experts ashore can tell us whether these are spiny, as in hard, or are they soft. You can make out their eyes though, tiny, just quite small eyes. So maybe they're more, they're very tactile animals, I guess. Down at these depths, it's completely pitch black, obviously.
And our fish expert John is saying this should be modified fin rays, so theoretically they're soft.
Yeah, thanks John. That's good. Modified pelvic fin rays and pectoral fin rays. So they'd be sitting on their pelvic fins, and their pectoral fins are behind the head, and that's contributing to these really quite long extended fin rays extending out from the head. Yeah, thanks very much there, J-Rod. Got J-Rod and Chris on the controls, and we've got to keep moving up slope. Yeah, we have seen tripod fish before. In fact, one of the earlier dives we did, we got just absolutely spectacular sideways look at a tripod fish. Quite amazing looking animal. So just manoeuvring the Falkor just ahead of us as we move up slope. Yeah, thanks, John. John's just John Pornowski from CSIRO is just saying the eye's probably not much use down in these depths, but it's certainly making good use of its fins as, you know, tactile sensors for anything drifting past.
Keep moving up.
Yeah, thank you. So we've got Cody and Chris on the ROV controls, and John on the wireless internet connections. John, can you bring up an inset of the inflator mass there, please?
If we see a question on that, we can just try to answer it.
Yeah. Yeah, just putting an inset view in here. The sort of grey-blue is what we've mapped off here off Cape York in the last 2 days. So it's been really spectacular mapping so far. This section is extending out from Princess Charlotte Bay, which is a very prominent bay along the coast of Cape York, and we're just zooming in on what we're calling Rodda Canyon, because it's basically draining just directly east of Rodda Reef. It's only the Great Barrier Reef, the shallow Great Barrier Reef, is only about 6 kilometres away from where we are now. But you have a look in the foreground. It's this big ridge that we mapped last night, and so quite spectacular seafloor. This was all unmapped until we arrived here 2 days ago. So we had no idea that there were these big, deep canyons in this part of the Great Barrier Reef, which for me is a huge thrill to be able to reveal these deeper parts of the Great Barrier Reef. Certainly the shallower parts of the reef itself are very well mapped through LiDAR, aerial LiDAR surveying done by the Australian Hydrographic Office. But in these deeper waters, still a fair bit of the deeper Great Barrier Reef is unmapped, and certainly the Cape York region, which is the very far northern part of the Great Barrier Reef, have had no systematic multi-beam surveys done. So this Falkor expedition is a real step change in our understanding of what the topography looks like. So we're just, John's just doing a bit of a zoom around. You can see this, if you look up on the shelf, it looks like an old river channel. In fact, that's exactly what it is. Now it's underwater, of course, but during last ice age, say 20,000 years ago, sea levels were about 100 meters lower, and that would have been an ancient river that ended right at the head of this canyon. So in fact, where we've mapped that grey color is about where sea levels dropped to 20,000 years ago. And then we're in this very big canyon. Oh, here we have a beautiful sea cucumber coming into view. One of these swimming sea cucumbers. Headless chicken, did you say, Cody?
Headless chicken.
Headless chicken monster, yeah. I might just linger here for a bit because they're beautiful to watch.
And all these sea cucumbers are so different from what we see in the shallow water, when all of them are trying to disguise themselves from their predators, bury themselves into the sand. As for the deep sea one, they're really colorful and they swim around. Yeah.
Yeah, different to the shallow water sea cucumbers, obviously. Like I don't see any those shallow water ones swimming around, but this seems to be quite common in the deep.
Yeah.
Yeah, I quite like looking at sea cucumbers. I know this is part of your project, isn't it, that you've
Yeah.
shown?
The project that I'm doing is for sea cucumbers that are very shallow. So they're all basically in the sand, reef flat, and they're so shallow when the weather is nice, and we just put a drone out on the reef flat and you take pictures on top at about 20-meter altitude. We have a clear image of what is below, and with the images that it would take, what I'm currently trying to do now is a very, very small pilot study to see the potential of automatic detection of the sea cucumbers. So if this one, because no one has ever been tried to use a machine learning tool to detect sea cucumbers in real life. Previously, in the wild. Previously, it's more for those sea cucumber farms, and they would like to have a tool to measure sea cucumbers' length and weight underwater to see whether it's a good time to harvest them. But the Grunpar has been thinking of to develop a tool so that they can have a constant long-term monitoring of the sea cucumber densities out on the reef in the shallow.
Big urchin, yeah. So, can you just put the lasers on, guys? And we just get a sense of scale here, please. There we go. That's a big urchin. Yeah, good 20, easy 20 centimeters across. Lovely. Yeah, that's a monster, isn't it? Yeah. So, it's an echinoderm. It's got spines, hard spines, but also has legs. And you can see, these are spines that we're seeing here. And in amongst them, there's probably tube feet as well, helping it move around. That is really interesting. These little cones at the end of the, like little snowshoes. If you're walking around on soft mud, helps to have some snowshoes. But you can actually see the tube feet amongst them. They're softer, more like a proboscis as well. And they literally using hydraulics, water, to pump and move all these spines and legs. It's pretty amazing. And of course, lots of these sea pigs. They're quite small. They'd be a good 1 centimeter across, but there's just hundreds of them. That is a beauty. On the expedition before this, when we're going up the Ribbon Reef sidewall canyons, we came across a big cluster of these urchins, and they were all moving around, leaving these very distinct bulldozed trails on the seafloor. We saw the trails to begin with, and we're wondering what they were, because they tended to be contour parallel, but then we came across a big cluster of these urchins, and they were just all moving around, sloping up the seafloor. There's another one. Maybe we've come across another cluster of them. Yeah, we're down at 1,634 meters, and it's pretty cold. It's 2.7 degrees Celsius. Yeah, okay, so we've got ourselves a little gastropod. Looks like it's got its foot out, and it's just moving slowly across the seafloor. kind of spiral gastropod. Do you know your shells at all there, Marty? It's a good size though. It's nearly 10 centimeters. So going back to your sea cucumbers there, Joan. So with the drone imagery, it's the pixels are sharp enough that you can detect species type of
For now, it's still on the whole Holothurian family, so it's not really on a species type, because most of the sea cucumbers that we find in the shallow reef flats, they do have a couple of size difference or a little bit of the feature difference, but it's not that obvious if you add up the water column on top of them, and also plus the sediments that usually covering the sea cucumber. It's a bit hard to tell apart.
But shape-wise, it can identify like that is a sea cucumber. Right?
We can identify that as a sea cucumber, but not to species level.
Yeah, okay. But the idea is to speed up the process of
Definitely.
identifying just from imagery alone.
Yeah, because from what I've done during the literature review part, previously most of the sea cucumber survey were done by manta tow. So a ship is a little speedboat in the water and towing a diver in the water or snorkeler at the surface, and usually they will cover.
There we go. That is nice.
It's another holothor, or that's a big eye.
Halosaur, perhaps. Reaching out to John, our resident ichthyologist. I think John's in Hobart watching. So, John, what have we got here? A rat tail, yes. Big eyes, very distinct.
Dorsal fin.
Dorsal fin, that's right. Thank you. Getting my fish bits right. Thank you. But look at the size of the eyes. Quite different to like the tripod fish, for instance. My guess is this one's trying to use as much visual cues as possible for hunting. John's saying likely Coryphanoides whiptail. Hope I've said that right, John. Anyway, we do really appreciate all the expert opinion coming from ashore. Myself, I'm a marine geologist, more focused on rocks, obviously, but I do quite like looking at critters, and I have to sort of reach back to my undergrad years from many years ago to try and remember what we're looking at. And we have Marty, of course, is just finishing her PhD. Had a bit of a celebratory champagne last night. Handed in her PhD thesis. Congratulations, Marty, Dr. Marty, soon to be Dr. Marty. Got Vicky studying Southern Ocean. Is it radiolarian diatoms? Radiolaria. Yeah, so very nice to have Vicky here. Vicky's she's going to be on board for 47 days, so we've got this is quite a big expedition. We've split it into 3 legs. So the first leg was from Brisbane up to Cairns, about a week, and there were 3 dives done. Picked myself, Joan and Valerie up. We're now up in the Cape York region, and we'll be here for the next 3 weeks. So just focusing on mapping this part of the Great Barrier Reef, the very far northern part. Then we will return down to back to Cairns. We will depart, myself, Valerie and Joan, and we get Luke back. Luke is joining us again from Queensland University Technology, and a few other people, some media correspondents and the like, and heading down into the Swain Reefs and then out into the Coral Sea Marine Park again to go and have a look at the Tasmanid seamounts, which are a chain of extinct hotspot volcanoes that stretch along the east coast of Australia, and they've got coral reefs on top of them. So it's a really interesting geography that we're going to be looking at through this entire expedition. So we're up on this, we're up on that plateau now. Yeah, it's definitely flattened out. All right. So is this a ctenophore? This looks like a ctenophore. Yeah, a deep water ctenophore. Just the sort of thing that Dougal Lindsay likes looking at. Very beautiful. You can see the, I guess, the iridescence, the rows of cilia, that run down the side of it, and when it catches light in all different angles, you get this. What do you call it? Well, it's an iridescence. So it's common to all ctenophores here. Now, if Dougal was, we got Dougal saying new species. All right, maybe. I think we better slurp this up. Yes, collect. All right. Yes, please. Yeah, Dougal's watching. Great to have you join us, Dougal, from Japan. Of course, we're actually having a dedicated Dougal dive a little bit later in this leg. For now, though, most of the ROV dives are going to be benthic, seafloor dives. But we will do a mid-water dive just for Dougal to watch in and commentate on a little bit later during this leg.
Does Dougal want the ctenophore collected, or does he just want imagery of it?
Dougal, question, do you want us to collect it, or are you happy with vision only?
Because
It's very dark, isn't it?
Just imagery is fine.
imagery is fine. We don't.
Yeah, okay, thank you. Thanks, Dougal. Okay, we're just getting a good look at it.
Sure. Yeah, they don't. Sorry, Chris. We'll find you something.
It's very beautiful to watch. Always reminds me of a UFO, you know, just hovering. Look at the colours sparkling up. Wow.
Do you ever see one that colour before?
Yeah, really dark purple. But yeah, if you just look at the sparkles along the cilia. You know, not even, like Christmas tree lights, with all the LED fancy technology we have kind of matches how pretty that is. Yeah, copy that, Dougal. We're just, because we're hovering, and there's a bit of marine snow around, probably kicked up from the sediment that we've kicked up.
So is this a range expansion, Dougal, or do you think this is an undescribed ctenophore? It is new. There you go.
Wow, okay.
So we need to get good images of this from all angles. And so, Dougal, if I remember correctly, we're not collecting the ctenophores because they don't preserve. Is that right? So we're better off getting the in situ images. Put tissue in a tube of ethanol. So you do want it collected? DNA to go with the image. Okay, so we do want to collect it, please, Chris. And Cody wants to have a good long look at the gonads. Easier to publish. All right, we will collect it.
Oh, that's a nice shot. That's beautiful. Wow.
That is beautiful. Well, we keep saying that on every dive we find something new, and has not disappointed. Just to cap off all the excitement that we had earlier. Test tube baby fluid. What?
Hermaphrodite.
It's hermaphrodite.
Maybe I
It's a
get it.
hermaphrodite. Wow.
Test tube baby. Yeah, right.
Okay. So this is quite deep.
Nothing's in two, correct?
Yeah.
It's two open.
Yep.
Yep.
So we're currently at 1625 meters water depth, and it is 2.8 degrees Celsius temperature. And our colleague Dougal Lindsay is saying that he's collected a related species from Japan in about 2,000 meters. But this looks like it's a new species, undescribed, and certainly this is the first time that we've observed or collected these in this region.
They got it.
Falkor.
Bathysauroi Falkori. So we'll just check the jar, make sure we got him.
There's the squat lobster with
From the
the
rubbish
trash.
collection. And there's the brachiopods that we got earlier. Seeing up.
At the bank. Is that it at the bank?
No.
No. Let's look through the tube.
Might be the grading.
Okay.
Go back to
Yeah. It's
there it is. It's in there.
That's in there, confirmed.
which tube?
So
there it is.
Yep,
Yeah.
yep, there it
Oh,
is.
look at the
Oh,
size
look at
of
it. There we
It's
go.
a beauty.
It is beautiful.
That is quite big, isn't it? Yeah, really fragile. Okay, so we're going to have to work hard to preserve it. How are we going to do this one?
We just need to get a small tissue sample and put it in ethanol for DNA. We won't be able to preserve the
Not
whole
the whole animal.
animal.
Okay,
No.
so just a portion of its
Yeah.
flesh.
Yep.
Okay.
And Dougal, is there any particular part of the animal that we should subsample for the tissue for DNA? Anyway, we can talk about that at the end of the dive. That's great.
It's huge.
Wow.
Yeah, fantastic.
Okay. There we go. Now the Falkor first. Very fragile. Yep. Okay. And we'll continue making our way up the slope, up the side wall of this canyon that we're in, Rodda Canyon, in the vicinity of Rodda Reef in Princess Charlotte Bay.
Great ROV, I started to move it again.
We're just talking about logistics. We're trying to limit our dives no more than 7 hours from deck to deck. But being fairly deep here, it's 1.6 kilometres deep, and our target depth at the end is about 1.3 kilometres deep. It takes us about an hour to recover the ROV, so we just don't want to push it too far late into the afternoon before we start to recover. So we're going to continue upslope, see what interesting things we find. Still plenty down here on the seafloor, including these whips that probably look like soft coral whips, and they've all had these brittle stars attached to them, with their arms intertwined around them, like epiphytes on a rainforest. So it's a pretty common feature in the deep sea where tall corals are used by other animals to cling to, like their own little habitat. So temperature down here is 2.7 degrees C, but talking about cold temperatures, we've got liquid nitrogen on board, haven't we?
Yes, we've got a big dewar of about 20 liters or more liquid nitrogen on the boat. This is for Jeremy's project. He's cooperating with some other scholars doing a 1,000 genome project. It's about this deep sea species, both corals, sponges, soft coral, hard coral, black corals. All of these, for certain of the samples were collected, we need to do a fast freezing preservation to keep those tissues, the DNAs intact, so we can send them back to do a whole genome sequencing.
That's right. So that's the key here.
Yeah,
Whole
that's the key.
genome.
Whole genome.
Yeah.
Yeah.
So when you preserve animals in ethanol, you may not get the entire DNA structure because it breaks down quite quickly. So it's great for preserving tissue, but can actually break down the DNA. Here we are flash freezing
Yes.
tissue samples. So we have a whole protocol set up, and we're going to do it for the first time today.
Yep.
Hopefully we do it all safely. But what temperature is this liquid nitrogen? Like-
That's a good question. I'm not so sure. It's definitely below -80 because our emergency protocol is, if there's anything wrong with that liquid nitrogen dewar, we will transfer all the samples into the -80 fridge.
Yeah.
So I would assume it will definitely be below that. And from all of our training with liquid nitrogen is that we need to wear all the PPE possible and never really stick your body, your hand or fingers.
Oh, is this
into-
an octopus?
Ooh.
Hey, we got another octopus. Beautiful, big eyes. Let's have a look at it. Oh yeah. Now, you remember the one we saw earlier
Yeah.
there, Joan? It was red. This one
It's more pale.
Yeah, pale. Big eyes though. Very not moving much. It's a bit starting to unwind its arms. Quite big. Oh, it's just trying to.
It looks like it's trying to get the jelly.
No, it's probably just wondering what some alien just landed in its backyard. All right, so if that oh, nice shot, beautiful.
Wow. Look at the
Oh.
texture on the.
Oh yeah, those eyes.
Yeah.
That is amazing.
Ah.
This is the SuBastian's lights looking at it. You see it reflected in its eyes. Just beautiful. The suckers. Yeah, we just, it's all quiet here, just in awe of this animal. The texture of its skin, you know, these little rough bumps, of course they can change them in an instant, you know, to match the texture of the surrounding environment or the colour, the ultimate in invertebrate. It's be able to change camouflage, colour and shape. Can you just put the lasers on briefly there, Chris, just to get a sense of the scale of this octopus? There you go. So the lasers are on, they're 10 centimetres across. So the head of this is at least 20, maybe, yeah, at least 20 centimetres across, maybe even up to 30 centimetres. So it's a decent size octopus. Beautiful. All right, well, we have to keep moving. We're about halfway along our transect up to the crest of this Rodda Canyon. So we started at 1,700 metres and we're heading up to about 1,300 metres. So about a 400 metre vertical rise over a 1 kilometre distance. And even though it's all soft sediment, there's plenty of marine life down there. Lots of bioturbation, plenty of little shrimp.
There's another
A big eel of some kind.
rat tail.
Rat tail, yeah. If John's watching, he'll be able to tell us. One of these fish with the big eyes. Another big urchin just off to the left here. We've seen a few of these already. Really decent size, you know, 20 centimetres across. With their little snowshoes on, those little white cones on the ends of their spines. Probably quite useful in this soft mud, don't you think?
Yeah.
Hey, Joan?
Imagine it's like when we're skiing on the snow, you always want something to balance the soft sediments underneath so it can move around.
Yeah. Oh, here's another one. It's ctenophore. I think it looks like one of the ones we just collected, don't you reckon? Very dark. Got this real dark colour. And if you look behind the ctenophore, you see these sea peaks. These are sea cucumbers, holothurians. Quite small. They're only about a centimetre or so across, and we've seen them all the way along this dive. I think they're the first. This is the first place we've really seen these sea
pigs.
sea cucumbers. Lots of them.
I remember we did carry one on the outside.
Yeah, John's saying Aldrovandria, so if I got that right, seems to be outnumbering the other fishes. That's good to know. We're really curious about the fishes in the deep, and yeah, I got what's the kind of assemblage makeup down here, and we haven't had deep water ichthyologists really kind of pull out these numbers yet, so that's really useful to know. Yeah, so we're going to continue upslope. We've had a few delays earlier on in this today for this ROV transect. We've got a ways to go, but we're looking at about another hour on the bottom here. Please stick around. Never know what you're going to find in the deep ocean, of course. So we've just learned about another hour down here, and then we'll start to recover the ROV. But the idea is to get to the end of this transect, which is at the crest of the of this particular canyon.
We really get a lot of the sea urchins.
Yeah, yeah. And I was just saying, at the Ribbon Reefs, which was the previous expedition, they left these real bulldoze trails, and when we first saw them, they were contour parallel, right? So they were running across the slope, and we didn't really know what was creating them until we came across like this big field of urchins, and they were bulldozing the sand in front of them, but not going upslope. They were definitely driving backwards and forwards, like leaving zigzag trails. Ooh, there we go, some outcrop. Pardon? Yeah. This looks soft to me, Marty. Soft. Yeah, yeah. It's got the dark staining is ferromanganese oxide, which means this face would have been exposed to water for a long time. It can stabilize it, you know, if it is soft. The ferromanganese crust, can actually, it lithifies, it becomes almost rock-like, but of course once you push into it, it's actually really soft.
Is that a prawn shrimp?
Just missed it. Yeah, yes please. Yeah, we have to keep moving upwards. It's an anemone. One of these really weird, whatever this is. I thought it was a sea cucumber, but be happy to be corrected. It pulls in these tentacles at the end. There's definitely a mouth like there. It's weird. Any ideas, Joan?
To me, they do look like a weird anemone because they seems like they're attaching to that base there. But just now when we were poking the base, they're not that hard. They're kind of like soft sediments.
Yeah, see how it's opening up now? Yeah, I thought it was a sea cucumber, but I'm not so sure now.
Dougal's suggesting maybe a tunicate as well.
Yeah, you know, like an ascidian generally has an in pore and exhale. Do you see up on the top here?
Yeah.
There's like a small pore on the top.
Yeah.
See that when he went to go open up, and then right before he went to fully open, it like exhaled some stuff at the top.
Yeah, okay.
So the bottom end right here got really, really small.
Yeah, okay.
Yeah, that could be
An ascidian of some kind.
tunicate, like Dougal suggested.
Yeah,
Yeah.
very weird. Good size too. We've seen a few of these now. A little shrimp behind them. Yeah, there's plenty happening on this rock. Well, we know it's not rock, it's actually soft sediment. It's kind of lithified. But then you get this ferromanganese crust, which makes it semi-hard, you know, enough for animals to attach to. Oh, big eel. What do we got here, John? And Luke's joined us. Welcome aboard, Luke. Luke had left us in Cairns, unfortunately. Sorry that you can't be here with us. Luke's coming back for leg 3 of the voyage. So anyway, you can enjoy it from ashore there, Luke. That is it's a great shot.
Yeah. John is suggesting this is basket work eel. It's
Beautiful.
apparently having a full stomach.
The stomach of what? What are they eating, John? This is a big question. There's a lot of these, you know, these fish down here. What is it that they're eating? There's lots of crustaceans. Do they eat crustaceans? Oh, see, here we go. This is a stalked crinoid or sea lily. This is one of these relict animals. I really like these. Only find them down in the deeper ocean. Of course, they're an echinoderm, but these are stalked. They don't move, unlike their other feather stars that can actually move around. These ones are completely connected via a stalk, and you find them in the fossil record going back hundreds of millions of years. So I talked earlier about this big limestone province west of Cairns called Chiligo, and you can see the broken up remains of these sea lilies in the limestone, which is over 300 million years old. So these animals would have been a really important fauna back in those early Devonian shallow seas. But here they are, living relics in the deep ocean.
Yeah. Can we?
And brachiopods. Fantastic. And more living brachiopods. Another one of these relict animals found more commonly in the deep geological fossil record. Sponge? Yeah.
Maybe Jeremy is waiting to have a look at this thing.
That
Yeah.
which thing?
The one you were just pointing at.
The little white
Yeah, the
thing?
little white. Yeah.
There's like a little polychaete worm here. There's plenty on this rock here. Now we've got squat lobsters, brachiopods.
That one.
Yeah. And just off the left, these look like comatulid crinoids. So these are mobile feather stars. They can move around. They can even swim.
So it's like the white featherish thing is the same as the orange crinoid that we.
Yeah, they're related. Yeah. So these are comatulid crinoids. They can move around, right? So you could pick it up in your hand, and you know, in fact, they can swim. They do this beautiful swimming motion if they want to escape. But the sea lilies can't move, right? They're stalked and they're attached. Ooh, it looks like a sponge to me.
Yeah.
Something that Merrick might like.
Jeremy is looking. Yes, he's looking right now.
It's a crab.
It's crab. What?
Or squat lobster.
Oh,
Wearing it? Wearing
it's
it?
one of these decorator. Yeah, a hat, a little sombrero.
Oh, so it's a sponge tissue.
Yeah,
Yeah.
yeah. You know, some of them they put anemones on the back. That's part of their
I've
defense.
seen the anemones before.
Yeah, this looks like Mexican sombrero.
This guy's got style.
Yeah.
In the yellow box. In the yellow box. It's a hydraulic pressure.
So, do you know what this is?
No,
No.
don't know. I mean, some of these are polychaete tubes.
Yeah.
You can see down the bottom right here. You can see the feeding tentacles from the polychaete. But if you look over to the right, this is a cup
Yeah.
coral,
Yeah, I think yeah.
and these are brachiopods. They're almost
Those are
bivalve-like.
Is this like also small brachiopods?
Yeah, some of these are cup corals, right?
So
you can
this
actually
is
see
cup-
the
yeah.
individual. They're solitary polyps.
Yeah.
They're a scleractinian coral. They've got a hard,
Yeah.
they got hard skeletons.
Yeah.
They're an
Yeah, this is quite obvious with the skeleton here and then a little bit translucent tissue and tentacles covering on top.
yeah, and we have been collecting them during the
Yeah.
expedition. We've got an interest in collecting some of these. But just to the bottom left, that'd be a very like a polychaete
Poly-
tube.
Yeah, there are a lot of cup corals here.
Yeah, that's right. There's another one here.
Yeah.
They like these overhangs.
Yeah.
You know, the overhangs are really great because there's the sediments not building up on top, and you know you get a lot of things connected to. All right, we're going to keep moving up past our sea lily.
Clean it up.
Beautiful. Yep, clean it up.
Smoky mirrors here.
Yeah, sorry about that. Off we go. Got to keep moving. We've got to watch our time too. We've got about an hour left before we have to pull the ROV off the deck. So we're 1.5 kilometers down. Temperature is 2.9 degrees C, and we're climbing up the northern sidewall of Rodda Canyon. So we're about 6 kilometers away from the edge of the shallow Great Barrier Reef. And this is an area that we've been mapping over the last 2 days. Quite spectacular canyon system. Dropping literally from the edge of the coral reefs, the shallow coral reefs, down to well over 2,000 meters deep.
That's deep.
And off we go. So just heading up slope. Still, it's quite steep.
There you go. Look, hang on. There's a nice rock.
Oh yeah, look at that. Nice. Okay, we do like rock. Rock being, in this case, is probably, it's quite, there's a lot of strata here. Be good to touch it to see how hard it is. We've got black coral down in the bottom right, and black coral up on the right hand side, and a sea lily. Just
Yeah.
there's a lot. Yeah, lots happening here. We should just have a bit of a linger.
Yeah.
Yeah.
Jeremy's getting very exciting. You
Yeah.
probably want a good look at this.
He's excited about us poking the rock. Good on you, Jeremy.
Yeah. We're converting Jeremy into a geologist here.
Is that something you can look at? I think that's a little bit too big. Okay. Could be if we can zoom in. If we can zoom in the blood just like this too.
Okay.
Yeah.
All right. We'll just have a look at the black coral first.
Lots to look at here.
Yeah. Yeah, you can have all these little micro ledges, each one of them their own little habitat. And you see, I can see brachiopods underneath some of these ledges. Soft. Yeah. Okay, so we're looking at a lot of layering happening here, but it's soft sediment layering, and it's got this a crust of ferromanganese oxide on the outside, just enough to probably lithify it, make it a little bit hard to allow these animals, these sessile animals like corals, to attach to. And of course, creating enough of an overhang, yeah, enough of an overhang to allow lots of animals to attach to underneath and not get buried in sediment.
Maybe that's one right there.
So will the sponge be interested for you, Jeremy?
It's probably of interest to Merrick.
Yeah, Merrick Eakins is our sponge expert from the Queensland Museum. He's interested in any of these deep water sponges. So we're just trying to find a good place to settle down here, so we can. So we'll be able to do some sampling.
Right arm is frozen. Okay. All right.
Yeah, so just to repeat, this isn't hard rock, you know, like outcrop type rock. This is layered soft sediments, but enough of a layering and exposure to the water to allow this ferromanganese oxide to coat it and just make it hard enough for these animals to attach to.
Is it possible to zoom into that overhang a little bit more?
Once we've got the sponge.
Yeah, that looks like a healthy sponge. That's the sort of thing that Merrick would like. See a little stalk at the bottom of it. There's a brachiopod just up here. There's quite a lot happening here. Little cup corals.
Are we trying to use the arm camera to have a look at the sponge, or this is collect?
This is a collective.
Really good picture.
Oh, misses. Yeah. With the situation camera, you see, that's why it's really useful to have both cameras.
Yeah.
Yeah, we got it. So that funny smoke light you're seeing is just where the one of the tubes is opened, and the water that we had in it, which was surface water, is of a different density to this colder, probably more saline water. And so as we open it, it completely changes the optics as the water mixes.
Can we have a look at the whip coral later?
Oh, I see.
Jeremy wants to look at the whip coral.
Yeah.
Okay. What are we going to do?
Also asked to try for a horizontal push cord.
Okay. Okay. Yep. Yeah, so we're going to do a horizontal push core. Most of the time they're vertical push cores down onto the seafloor, the flat seafloor itself. But here we can, Sebastian's ever flexible, and we are going to push it, try and push it through this strata to get a sense of what we're looking at here. Okay, so we're just organising the push cores here to. We're going to.
Oh, sorry. No, you're right.
And we're going to see if we can push it into this vertical face. That'll give us an understanding of what this sediment is about. We can do lots of things with it. We can look at the layering. We can look at grains. We can even do radiocarbon analysis. So there's quite a lot we can do with all of the sediment that we collect. There we go. Whoa, okay. Okay. Yeah, recovery wasn't so great there, I guess. It kind of just disintegrated. It could be that it's just too difficult. We've tried grabbing it before, you know, with the claw hand, you know, literally grabbing a piece. But that might be our fallback option. Yeah, definitely. This is a core sample. So the beauty of a core sample is it tends to preserve the layering of the sediment. But yeah, this is probably, it's too lithified to do it this way. Anyway, you've got to try a few different things just to see how it works. Okay, so it didn't work. I was saying we tried to claw a little piece before, but again, as soon as you put a bit of pressure on the rock, it just disintegrates. So, but we may have another try at it.
That's two huge lobsters.
Oh yeah, yeah. I didn't see the one behind it. Yeah.
Particularly interested in the white corals that are around.
There.
Yeah.
That's that one.
So you can see the Sebastians, the guys are clinging onto the rock just to keep it stable. couple of brachiopods just above it.
Yeah.
Actually, there's quite a few brachiopods. So have a look how many there are underneath here.
It's a little bit hard to focus on such a thin branch, but the key of identify a coral is always try to look at its polyp.
Yeah, that's right. This is really small. So you're going to try and do manual focus here so we can have a closer look.
I think Jeremy right now must be.
So we're
poking his
thinking
head.
black coral?
I believe so.
Has it got this double row of... It's very delicate.
Yes, let's grab this one. It seems to be Stichopathus, which is also one of Jeremy's target species.
Yeah.
Is he gonna wanna put this in a quiver?
Yeah.
And
So is it flash freeze or just ethanol?
let's double check. Do you want it in a quiver? Not in a quiver, in normal ball box.
Yeah. So it's an ethanol.
Yep.
Ethanol only. Yeah. Still, the one underneath it's pretty spectacular.
Hold chair over there, slowly. What's that?
There we go. Whole colony. That's good.
Nice.
Yep. Perfect.
Wanna go into
See.
the very bottom?
3-4.
You said 3-4?
I think it's too big to put in C.
Is it too big?
Put it up to the camera.
Yeah, can we bring it close to the camera to have a picture of it?
Definitely.
So we've got Valerie on our event logger. It's one of the important seats in the control room, and this is how we keep track of all the samples we take. So we take photos, event photos, where the animal is in place, and then we bring it towards the camera so we can take a photo of it there, and then we also take another photo of it when it goes into the box. So by the time it comes up to the surface, we know exactly what animal it is, so we can track it all the way through to the time we preserve it and back on board the ship. So it's quite important to have that event logging happening all the way through the collection process.
I'm still wondering what's that little nodule attached to this piece.
Yeah, there's maybe some eggs of some kind, you
Yeah.
know. Anyway, we'll find out soon
Yeah.
enough when it comes up.
At the beginning I thought it might be like a teeny tiny cowrie, but now since it's moved, it doesn't look like one anymore.
Yeah. Well, we have seen shark eggs, like the mermaid's purse, attached to some of these long whip corals.
Yeah.
That's always pretty amazing to see. I don't think that's certainly not the case here, but
No.
but you know, animals do use the stalk of the colony to attach eggs to.
And Jeremy also says it's pretty common for anemone sometime to attach onto a piece of whip coral.
Yeah. Okay. All right. Well, we'll find out.
Yeah.
We'll be able to tell you whether it's an egg or an enemy or something,
Yeah.
and some other colony.
Feels like all these bio box, it's Christmas gifts. You just come back up and unboxing all of them and see what is inside.
Yeah.
Exactly.
Yeah. That's right. Yeah. Yeah, we have had some pretty amazing marine life collected. Over the, this is the third expedition to Great Barrier Reef and Coral Sea. What are we talking? At least, nearly 3 months worth of Falkor time. This current expedition, the northern depths of the Great Barrier Reef, is 47 days long. So it's quite a long expedition, broken up into 3 distinct legs. So first leg was from Brisbane to Cairns, over about a week, and 2 dives were done. And this second leg is for about 3 weeks, up in the Cape York region. The very
These two. So
This one?
I think both. Like maybe which one is easier to go first?
that looks more live.
Yeah.
Focus on that one.
Oh,
yeah.
Yeah, so we hope to do a lot of ROV dives.
Can we just zoom in first? Yeah.
Just to let you know, the plan over these 3 weeks, we want to do a lot of mapping. This far northern part of the Great Barrier Reef has never had any systematic deep water mapping before, so this is the first time. It's also never had any deep ROV dives done, certainly not this far north.
Can we look at the one to the right now?
And now we're just checking some of those look like whip coral structures attached to the overhang of this rock. And this. So is the camera auto-focusing right now most of the time? No,
Manual focus, yeah. Most of the time it's auto, but when you get close like this, it's better.
Yeah, this
manual.
looks like a dead one with sediments on it. Yeah. I think we can.
Yeah, so these animals that are being flash frozen into liquid nitrogen, yeah, we are looking at the endosymbionts DNA, but also the host DNA. We're trying to, my understanding of the project is they're trying to understand the DNA structures between the two co-evolved animals that are using each other, the host and also the symbiont. Is that right, Joan?
I think for a lot of genome study, this is definitely a part that they're trying to look into. Because, for example, previous study that I've known about is in the shallow water coral. They're trying to look whether the microbiome, not only the zoosynthetics, not only the symbionts that lives inside, but also the one that lives outside at the surface of their tissue, and seeing whether their adaptation can either assist or reversely affect their tolerance to heat or ocean acidification. So it's always interesting to see how these organisms are working together, whether convergently or divergent.
Yeah. So the DNA analysis help
Yeah.
understand, I guess, this connections between the two.
Yeah, definitely.
But to do that, you have to sample correctly and preserve correctly. Otherwise,
It is very
you know,
important.
yeah.
Yeah.
So this is going to be an interesting afternoon. It's our first
Very.
time that we've done it for real.
Yeah.
You've gone through a bit of a dummy run, have you? Back on Townsville?
We did, yeah. We were like trying to do a little bit rehearsal with no samples, but just try to go through everything in our mind.
Is that of any interest to Jeremy?
And is this interesting to you, Jeremy? And he's all good. It's similar.
All right, we might get moving then.
You take what about the rock?
Yeah, if we can invite it himself.
Current, hold on to the mast.
Alright, I need to try to get some sort of.
I'm just having a look up at it, and as soon as I break it off, then we're gonna.
Yeah, so the samples that are going into this genome project, they have their own special quiver, we're calling it.
Yeah.
It's a box that's separate from the usual sample boxes, so we are not cross-contaminating
Yeah.
the samples. They're kept in their own box. When the ROV comes up, they're the very first ones that we collect out of the ROV. We can leave all the others in the standard bio boxes until we get round to them, but the important thing is that we really take good care of those genome samples so that they are flash frozen very, very quickly.
Yeah, because that was from Jeremy's past experience when he was doing DNA sequencing. He found out even the samples has been in the bio box for a short amount of time, but just the time when we are strapping down the ROV, trying to make sure all things are stable, the DNA are still degraded.
Yeah.
Yeah. And I did a similar project before with someone at AIMS, so Australian Institute of Marine Science. They are looking at the symbionts that are living, the microbiomes living on the surface of coral and sponge tissues. Those are also, at the same time, when we take it out of the water, pretty much the same time we're going to put it into the liquid nitrogen.
Yeah, so we're going to be real quick about
real
this.
Quick ones.
Yeah. I think it's going to be hard to get a rock sample here, Marty. It's all just disintegrating. All right, I think we'll just get going. We've got half an hour left on the bottom here, and we're at 1,550 meters, 3 degrees C. We're trying to get up to around about 1,400 meters. So we've still got a good 100, 150 meters vertical height to get through. And pretty steep. There we go. Some feather stars, comatulid feather stars. These are the mobile ones. They can move around. They've got little legs. They can cling onto the ground, like
substrate surface.
Yeah, on coral reefs you see them a lot.
Yeah.
You know, because these are deeper ones. They look a lot more delicate. But you know, at night time when you go scuba diving at night on coral reefs, I often see the feather stars coming out.
Exactly. Have you seen one of them freely swimming in the water before?
Yeah. They have this beautiful, you know, sort of
Dance.
it is like a dance, isn't it? Yeah. Yeah, I've done a fair bit of work in Antarctica, and down there's quite a lot of feather stars, comatulid crinoids, and I remember we had a deep sea camera down on the seafloor, and as the camera was drifting over the seafloor, they would all jump up in the water column, and so there was this, you know, we're talking hundreds of them, all of them swimming with that beautiful graceful motion. So probably the best place I've ever seen for swimming crinoids.
Is it all colorful ones or?
No, they tended to be quite dark, but they were big, and lots of them. So I did some work with Marc Aleum from the Paris Museum of Natural History, and he and I were on the Aurora Australis together, and he's a world-class crinoid expert, taxonomist. So it was great working with Marc. But you know, temperatures, it's very, very cold. It's Antarctic intermediate water here. The water masses were created down in the Southern Ocean. This is the water mass that we're in at the moment. Very steep. Feather star coming into a sea lily
And
coming into view.
Coral, another maybe soft.
Yeah, but, if it were closer, it might be. I'm guessing it's a bamboo coral, likely.
If that's a bamboo coral, Jeremy wants to maybe in a quiver. Yeah.
We'll just have a closer look at this one. Ah, right. Oh wow, look at the. Yeah. Right, we're just looking at the situation camera, which is gives us a wider view. and we're seeing turbidites. These are very soft sediment, mini avalanches essentially, and they layer up, and we're looking at crossbeds. So if any sedimentologists out there, that's crossbeds is something you'd understand. Just looking at a bunch of different cameras here. We've got this beautiful science cam view. Sea lily down on the bottom right, and looks like a black coral right in front of us. Yep, and Jeremy's interested in that.
And we don't, yeah, oh, but we don't need to take it.
But we don't need to take it.
You don't need to take
No.
it, just looking at it.
Yep. Okay, we're going to... That beautiful sea lily. Okay, we're just trying to stabilize the ROV here. We want to try and get a rock. There's quite a view in the other camera. It's directly above us, but it's so steep here.
This is so soft.
Yeah, yeah. Okay, so this is sort of classic turbidite. Lots of layering. But pretty hard to get a sample. It just tends to disintegrate. Oh, the whole thing's loose.
Sure you don't want a big one?
Yeah, we can take that. It's breaking up.
You can see the tiny feet that is attached.
Yeah. Oh, we're losing it.
Do you want that stuff thrown away as well?
Yeah, we can take that. We can have a look at it.
You can.
Oh. God, it's so soft. Yeah, well, this is we've been trying all day to get a rock, because it breaks down, it just falls apart. These are layers of turbidites, so very fine sediment that basically pulses of soft sediment that, like little mini avalanches that roll down slope, and then they stabilize and become another layer. So what we're looking at is layer upon layer of turbidites. But the actual material it's made of is a very fine grained mud. it's quite a delicate balance, isn't it? You need enough pressure to be able to grip it, but if
But
too
not too
much,
much to crush
and
it.
it just disintegrates. There we go. That's a better piece. Okay, we're just going to continue moving upslope, and directly above us, we've got a really big face of these turbidites, these layered, these layers of fine grained sediment, these little mini avalanches, including cross beds. So imagine if you have layer upon layer. Each layer sort of building up like a dune, and then you take a slice through it, you can actually see these cross beds. There we go. So we're going to head up, continue heading up. So we're at 1,536 metres, heading for around 1,400 metres. And there's those cross beds I was just mentioning. So you can imagine each one of these is a layer, a little mini avalanche slope. And when they come down, they can stack on each other, prograde forward, and you end up with this cross bed effect. So if you train in geology or sedimentology, this is a sort of layering that you look for that indicates that sediments have built up in almost like a dune effect, right? So one layer builds up in front of the other, but when it's exposed like this in a big steep wall, think of a quarry face where there's sedimentary rocks built up, you can get this crossbed effect. Very pretty. We've got all kinds of things here. Yeah, we got comatulid feather stars, we got stalked feather stars, and this really interesting, I don't know, is that a soft coral?
So there's a carnivorous sponge.
You want the sponge?
Yes, we do, thank you.
What's this?
It's a sponge.
Yeah?
I think
Yeah.
Crazy.
So, Jeremy, is this a collection for you, or otherwise we'll collect this for Merrick and keep it separate to your samples?
Just
he's still typing.
starter right now.
Merrick. Okay, great. Thank you.
You're saying that is a sponge?
Mhm.
Wow. Crazy.
Yes, we definitely collect that. Thank you.
Where's it gonna go? Two. Yeah, what's our bio box? Sure.
Yep. Absolutely.
Yeah, I thought this was a coral colony.
Yeah.
Carnivorous sponge. Well, there you go.
Is that our fight?
Chances are it's something new.
Very strange.
Yeah. Jeremy said he collected one of this last trip.
Okay.
Yeah.
Rod, can you, when it's stabilised, could you just zoom in? We have a quick closer look at the branches.
Trying to get a really good picture here.
There's a bit of sediment around.
Just chilling, sitting right there.
It's quite different, isn't it, to
There's
a
no
typical
skeleton.
coral colony. It's just
Yeah.
very different.
Yeah, I guess to me the most different part is the transparency, and you couldn't really see any skeleton stuff, similar things underneath.
Yeah, well, the sediment. Yeah, one of the questions is what's the sediment made of? It's a hemipelagic mud. So part of it is sediment that rains down as plankton particles, you know, bits of foram and things like that out of the water column, but also fine mud, which is that drifts off the land right into the Great Barrier Reef shelf itself, and flood plume events and things like that, which happen mostly during the wet season. Just mobilises all this very, very fine grain mud in freshwater plumes, and just spreads across the Great Barrier Reef and out into the Coral Sea, and eventually that all just accumulates, and hence we call it hemipelagic. Part of it's pelagic and some of it is this fine scale mud, and it accumulates as this very fine mud that at times in the past have mobilized like a little mini avalanche, like a thin, fast moving, partly fluid, which we call turbidites. And over time they've built up, as we're seeing in front of us, and now the face has been exposed, eroded, and so we're looking like at a cross section of multiple layers of these turbidite deposits. But if we were to actually look at the sediment in a microscope, we would see a real mix of different kinds of things. Very fine scale foramnifera, planktonic animals like single cell organisms, but also a lot of it would be mud, like a really fine mud. Well, it's so fine that you could barely feel the silt if you actually rubbed it between your fingertips.
Just keep going.
Just keep the camera on that rock faces and go up.
Yeah, we've only got about 10 minutes. So let's just go full drive and see what we see there. We've only got about 10 minutes left here on the seafloor, so we're just going to continue driving up slope until we call it quits. We've still got about a good hour to bring the ROV up to the surface, and it's getting a little late in the afternoon, so probably about another 10 minutes of live video time, and we'll just continue up slope and see what we can see. So just talking about the plan over the next few weeks, The Falkor is of course doing a lot of mapping, a lot of seafloor mapping. That's the main focus of the expedition, is to systematically map this offshore Cape York region, which is, we consider it as a frontier part of Australia. Certainly on the land, very few people go up there. There are no big cities or anything like that. But in the offshore marine area, this far northern Great Barrier Reef, it's also similarly very rarely visited by even tourist vessels, and hence we know very little about the deep offshore area. So over the course of the next few weeks, we're going to be mapping. We're going to be working our way northward, trying to 100% map the seafloor from the edge of the shallow coral reef down into the deeper depths to around perhaps 2,000, even 3,000 meters depth. We're going to find lots of canyons and interesting seafloor features. About every 2 or 3 days, if the weather allows, we're going to try and do an ROV dive. So stay tuned. We're going to be doing as many ROV dives as we can squeeze in during these next 3 weeks, these next 2 and a bit weeks, and so interspersed with a lot of mapping. So there's lots still yet to come, and it's been a pretty good dive, I would think. Our first dive for leg 2, our 4th dive for this expedition, the northern depths of the Great Barrier Reef. Okay, we're going to extend a little bit further, about another half hour on the deck. We're aiming to get to the crest of this canyon. It's around about 1,400 meters, so still got a little ways to go in a vertical sense. Looks like we're going to extend a bit longer. Oh, big fish over on the right, big eel.
John's saying it's another basket work eel.
Basket work eel or cutthroat eel. Yeah, I do like these turbidite layers. You get lots of overhangs. Oh, big sea star on the right. And a black coral above it. And an Echiuran worm. See a little Echiuran worm in the middle? It's got this, yeah, if we were to touch that, it would disappear in its hole. It's just, we've seen these in lots of places earlier. Not this dive, this is the first one we've seen on this dive, but in other ROV dives, you get these Echiuran worms. The actual animal is sitting in a hole, and it puts out these very long proboscises, that word.
We saw that quite a lot in the shallow when we were diving as well.
Yeah, and so the food lands on the proboscis, and I'm not sure if they pull it back into the body to eat it, or they somehow move the food down the proboscis back to its mouth. But
I'm not sure about that either.
yeah, but you can definitely recognise them because they've got this sort of Y or T-shaped proboscis that lays out on the seafloor. Yeah, lots hanging to the ledge here. Lots of quite delicate comatulid crinoids, squat lobsters. There's plenty to look at here. Brachiopods.
Jeremy, see that?
Yeah, definitely. I think now we're trying to zoom in to see whether that. Just want to copy the name.
Any we want to see what it is.
Yeah,
We're
it
just
looks
waiting for
like
it to
the
clear.
Gorgia.
So is that a zoom in and look at, or is it a potential collection?
I think it's a potential soft coral. The Gorgia Jeremy wants to have. Yes, let's get it. Let's put that one into the quiver.
Into the quiver.
Yeah.
Okay, so this is going to be one that we will flash freeze with the liquid nitrogen when we get it on board. Just see that there's a question on the YouTube chat about the basket work eels. Why are they called that? Oh, there we go. Thank you, John. I was just going to ask if one of our fish specialists is on there that can answer that question. The basket weave pattern on the skin is how they get their name. Okay. What are you chuckling at?
Jeremy said he's all good for this dive. He's very happy, and he will sit, and we can move on.
Oh, so he doesn't want this now?
No, he wants this.
He
but,
wants
like,
this,
after
and then-
this,
and then
he
after
was
this
so
we can-
happy, and he's just
Okay.
like, yeah, he'll enjoy the rest of the dive.
Excellent, excellent. Yeah.
I'll be able to get it.
Oh, did we get it already?
No,
no,
not
we're
yet.
just opening
Just
the
opening
quiver.
the quiver now to get ready to put it in. So the samples that we collect to snap freeze in the liquid nitrogen, we're keeping them in a separate container to all of the other samples. We want to try and maintain their temperature, so we don't want to be constantly opening the lids of the bio box. So that's why we're not putting these in the bio boxes.
Is it possible that we get a very zoomed in image for that, so we can have a snapshot? Yeah, have a snap image for its natural condition.
Okay. And that's another one of the
Yeah.
advantages of being able to collect using an ROV, is that you can get the surrounding context of where the sample came from.
It even has a tiny shrimp or a little squid lobster
yes.
there. It is really, yeah.
So we want him too, don't we?
Or yeah, I think we want them at the same time.
Yeah.
Yeah.
I mean, hopefully they come together.
yeah, hopefully.
Great. Yeah.
Yep.
Okay. That's good. Thank you. So, oh,
Oops.
hopefully our little shrimp comes with us.
Yeah.
Got that.
Hopefully we get the shrimp with it. Yeah, I think it's still on. I can see
think it's
the
in the-
eyes.
Oh, great.
Yeah, the little orange there. Yeah.
Fantastic. There you go. Get the lid on that, and then that's another sample for the genome
Yeah.
project.
It is always interesting to see these little critters whenever they're choosing a type of coral or sponge as their host. They will just co-evolve with similar patterns so they can
yeah, and
camouflage
colours
in
and-
it. Yeah.
Experts at camouflage, aren't they?
I think it's always like a diver's goal to go through like crinoids or this type of sponges or little coral branches and to see if there's a little critter on it.
Yeah,
Yeah.
yeah. Yeah, we like looking out for pygmy seahorses on the
Yeah. Definitely.
I agree. Good job. All right. Great. All right. Jeremy, you can put a blindfold on for the rest of the dive now. No, I'm only joking. If you do see something amazing, sing out. There we go. Wow. Super fine, isn't it?
Yeah.
Okay, up we go.
Great job. Yeah. Okay, wait.
We're enjoying the chat on the YouTube. About how much they're learning about random things like skin patterns on basketwork eels.
And also people saying they need to get a new pair of glasses.
Yep, we're all learning at the same time.
Circuit training.
Circuit training time. There's another one of those tunicate slash
Tunicate,
whatever.
Yeah.
So we've seen them at quite a depth range.
Yeah. From 17
From the bottom,
to
which was 17 to
yeah.
15, 200 metres depth range, we've seen them.
I think it's often that we see one of them and there's just like multiple very close by to each other.
Are you watching your messages just in chat? I'm just looking for messages from Jeremy at the moment. This is okay. This is fine.
I think Jeremy's all good for this dive.
Yeah, he says that.
Yeah.
But
Okay.
he's in the last 5 minutes when we're just about to break off from the end of the dive, he'll go, oh, just one more.
Okay. Noted.
And chances are it'll turn out to be an entire new genus, let alone species. Should we slow down and look at that? Yep.
Jeremy said he will only message if it is amazing.
Well, we'll just zoom in and look, and we'll wait. We'll stand by waiting for a message to say yes, it's amazing. Yeah. I think there was something just above it as well. Yeah, that one. Oh, what's the one above? Interesting. Okay. So Jeremy, even if we are satisfied with collecting, maybe you can still help us just ID what a few of these things are.
He's just really happy it's a soft coral.
Soft coral, okay.
Yeah.
Nice.
Yeah.
All right.
But we've already quite full bottle box, so it's
Yeah,
alright.
they are.
We have more dives coming up.
Yeah. All right, let's keep moving up.
Is it a big cup coral? Anemone?
Yeah.
So are they Nidaria? They're
Yeah.
Nidarian.
Yeah, they're related to, they're similar to anemones, but this is their own type of animal, definitely corallimorph. Yeah, so Mary pointed this out to me, and I've never forgotten it, because I was going, oh, what is that?
Yeah.
I think we got a little bit more soft coral coming. You can really see the
Something
eight tentacles clearly.
Yeah, but you see the stalk above it, looks like
Yeah.
something's attached. Maybe little eggs, possibly eggs.
Oh,
Nice rock.
can we grab those?
Chris?
Is that right?
See how that's
Yeah.
just sitting there like that?
Yeah, yeah.
Please grab that.
Grab it without touching it. Yeah, so this is a corallimorph, a deep water corallimorph related to anemones.
And
Are we going to
when-
try and grab this rock? It just looks like it's just dropped off
Yeah,
the
looks like
overhang.
It's just broken off from just there above it.
We haven't had a lot of success with grabbing these turbidite deposits. Because they're lithified to the point where if you try and pick them up, you can overdo it and just and crush them. But this one we might be able to get a sample of it.
Yeah.
Yeah, I see that.
Not quite a rock yet. Not cemented.
Yeah. Yeah, I once ran a project on some of these types of rocks, and they're at least 50% calcite cement.
Yeah. We call it indurated. Sort of slightly indurated.
Yes,
Kind of just
yeah.
holding together enough to be cohesive, but not yet lithified, which is when it becomes. Then we'd call it a rock.
Yeah, that's right. We found a range of them. So we had nodules that were completely rock, where the calcite was so developed that it was just a hard rock, you know.
Yeah, that's awesome.
Through to some that was semi-soft, like a hard, like a squishy mud.
Oh, sorry, I didn't make you do that. Opportunistic, right? Did you want to look at that? Any relative?
No, we're good.
No, we'll keep going. No, keep moving up slow. We don't have a lot of time left, but we're going to keep moving. We're nearly at, well, still probably got about 100 metres to go before we get to the crest of this this sidewall. So we may as well just keep moving along.
Yeah.
Yeah, so getting back to what you were saying before there, Marty, the rock is all different
What is this?
levels of lithification. There's that, yeah, carnivorous sponge
Is that
again.
another carnivorous sponge?
Yeah, it looks like it.
I reckon it is,
Yeah.
and I reckon we should take it. This group of sponges, this carnivorous sponges, are just. I mean, it's relative. It's all relatively new. Every sample we collected on the previous voyage was a new.
Collections, as far as I'm aware.
Yeah, that's right. So there's every possibility that any sample that we collect from here is going to be new. If not an undescribed species, then at least it's a new collection for the Queensland Museum.
Yeah. So on the expedition before this one, we focused on the area just off Cairns, between Cairns and Cooktown. So that's a good 300 kilometres south of where we are now. But as far as we know, no deep water sampling has happened up here in the very far northern part of the Great Barrier Reef. Certainly there's been a lot of work done on the shallow shelves around the shallow coral reefs themselves. But out in here in the deeper waters, deeper offshore waters, there's been no sampling. So this is the first time it's been done.
Do you want it?
Is there a box too? Or does it need to be on the back?
So as far as the mapping data goes, in keeping with the philosophy of the Schmidt Ocean Institute, all data is made open and public, and particularly the seafloor mapping data. So we have a project running with Oz Seabed Group, which is a collection of government, scientists, defence, industrial and academic ocean mapping people. It's been running for a few years now. Its director is Kim Picard from Geoscience Australia, and every day we upload all the mapping data that's been collected from the previous 24 hours to the Oz Seabed site. And they have a workflow that does processing in the cloud with the aim to have all of the seafloor mapping data published and available online for people to download within a few weeks of the end of this expedition, which is in late November. So probably around early December, all of that seafloor mapping data will be fully publicly available through the Oz Seabed Marine Data Portal. And of course that same seabed data is feeding into the Seabed 2030 project, which is run between GEBCO and the Nippon Foundation with the aim to map all the world's oceans by the year 2030. So this being a very long expedition, 47 days, it's broken into 3 legs. So legs 1 and 2, we have the ROV team on board. At the end of leg 2, the ROV team is, they're all going home for a well-earned rest. So that's Cody, Chris and J-Rod. I think Chris has been on board since, what, January?
They go home for 16 days.
Yeah, so Chris has been on board on Falkor since January this year. Definitely, everyone needs a break, and so the ROV team are getting off in Cairns in 30th of October. The Falkor will continue on as part of this expedition, but it'll only be mapping. So the ROV is staying on board, the SuBastian is on board the ship, but the ROV team is going home for a holiday.
That just struck my mind.
My to-do list is ridiculous.
Anyway, we hope to do a fair few dives over the next few weeks. We're trying to avoid back-to-back diving to give everyone a bit of a rest. So there will be a lot of mapping, but the aim is we'll map an area and fully, 100% map it, and then plan some dives, 1 or 2 dives, and then move to the next area. Keep moving northward up along Cape York. Remember, it's never been systematically surveyed with any kind of multi-beam mapping system before, so this is a first. And certainly there's never been any deep ROV dives, so we want to find out what's down there, what the rocks are like, what they're made of, and of course all the marine life that's down there. And the sampling is a big part of that. So over the next 2 and a bit weeks, we'll just continue to keep mapping and diving, and hope you can come along for the ride.
Chris, Unknown Explorer, says you're hardcore. And you must be part ROV. What's that thing?
Cup coral, maybe? All right, just keep moving up slope. Thanks, gents.
Wants to know what it is.
Yeah.
Cup coral. Cup coral.
Lots of turbidite layers here. It's pretty steep, isn't it, Marty?
Yeah, yeah. This is quite fascinating.
Yeah.
Okay.
See if we can inset in.
Yeah. Yep. We're full.
So just showing an inset here. This is the Fledermaus, the 3D visualization software that we use on board. The shallow coral reefs, we've coloured them rainbow colours. Red is really shallow. You could scuba dive on that. The area that we've mapped is this sort of browny grey colour. Just mapped that in the last 2 days, so it's it's amazing how much you can cover. And
It's the Tiedemann knoll.
Tiedemann knoll
That didn't exist prior to 24 hours ago.
Yeah, that's right. No one knew about that knoll, that really big bump on the seafloor. Yeah, thanks. It's sort of in the lower left there. Very odd. We cannot explain this. It's likely that'll be our next dive. Our next dive we're planning in a few days' time. Oh, look at these
Those,
ascidians, perhaps. And so you can see this is quite a big area that the ship is mapped, and that's a good 30 kilometres across the view there. And we're just zooming in very slowly onto the Rodda Canyon. You can see the canyon is draining. There's quite interesting head. The head of the canyon cuts right into the Great Barrier Reef shelf. Quite a narrow canyon, but it opens up down around 1,000 meters or so. It opens up, and we're on the northern sidewall of it. So we're actually trying to get to the top of it before the dive is finished for today. But it's been very interesting. We started down in the axis. The floor of the canyon was very flat, quite muddy, and lots of vegetation. We saw
that's right.
mangrove seeds, coconuts, big logs. Unfortunately, quite a bit of plastic.
Yeah, there was sargassum algae.
Sargassum, that
in the shallows.
It can only grow in the shallows. So the canyon is clearly draining. What is being washed offshore from the Great Barrier Reef shelf, and it ends up in this deep floor environment. And then for most of the dive, we've been going up the sidewall, which is really, really steep. And as we've discovered, a lot of this steep area is just these turbidites, these layered sort of mini avalanches building up one upon the other to create this layered texture that we're seeing. And because it's so steep, it erodes, and we're seeing the exposed faces of these different layers. There we go. We're just doing a nice zoom in. So we started at the... Whoop, we've lost it. All right. That's what happens when you zoom too close. But anyway, you get the idea. It's been fascinating to be here and just see, in reality what that mapped area looks like.
Yeah, thanks, John. It is incredible detail in that, here we go, peek around the corner, in that multi-beam data, the resolution, the quality of the time here, and we're mapping it real.
Yeah, the crossbeds are these sort of oblique layers. So if you can imagine almost like dunes, one layer adding a little bit more to the dune, and then if you actually cut through, like a cross-section through that dune, you end up with this classic crossbed.
This is amazing.
So steep.
Yeah.
I guess this is the thing with the multi-beam data. You know it's steep from that that sonar mapping data, but it's not until you put a camera down you realise just how steep it really is. Yeah. So David's asked, do these turbidites eventually form sandstone? More like mudstone, right? It's very, very fine grained. But yes, this is how sedimentary rocks evolve through time. They start as soft sediment deposit, and through various processes become solidified into hard rock. Oh, this is nice, isn't it?
Beautiful. Yep.
It looks like an old dead coral colony, and then there's just. Can we just get a little closer to this? Yeah, we got Chris and J-Rod on controls. Just this lovely little collection of feather stars. We got a sea lily or a stalked feather star at the end of it.
Yeah, it looks like there's
and it's a hexactinellid sponge.
Yep.
But there's at least 3, 4 crinoids, feather stars, sitting on a siliceous sponge. And those little orange things, I bet they're either polychaetes or some sort of crustacean living in the hole Oh, look at this coming out in the background.
Unbelievable! Wow.
Lots of sea whips.
I'm laughing
That's
because
nice.
everything always happens in the last 5 minutes of the dive.
Yeah. That is,
This is
beautiful.
spectacular.
Lots of colour here.
Yeah.
Beautiful. Yeah, if we can just have a bit of a drift past this,
Yeah.
that'd be lovely.
I'm having a little pinch myself moment here at the moment.
Yeah,
Can't believe I'm
we
sitting
do
here
get this
watching
occasionally where you just come across these clusters of coral just burst out of the wall. It's beautiful.
Yep, always happens right at the end.
Yeah, so this is a Brisingid sea star, this orange one. So it's not a feather star, it's a sea star. And they have these really distinct orange coloured arms. And again, when you get clusters of them together, it almost looks like a garden of flowers.
Yeah.
And siliceous sponge just above it. Beautiful, it's a beautiful sea lily.
All right. 5 more minutes.
Off we go. We haven't got much time left, so let's see what we can cover in 5 minutes. Bits of breaking off here, Marty, and rolling down slowly, sort of nodules.
Did he just boot that?
Distracted.
Shouldn't boot. Well, it's been an eventful day
Yeah, it has.
in more ways than one.
Yeah, we had that slight technical problem, sirens going, and everyone jumped back. But we're good now.
Yeah.
We're good. Fingers crossed everything goes well. We intend to do, well, a fair bit of diving over the next few weeks.
This is for the rock video. Oh, look at this. Wow. Just got a glimpse in the
Yeah,
cam
clearly
of
the
what's
overhangs
coming
make
up.
a big. Yeah, look
Yeah.
what's going on under the overhangs. Lots of bioturbation going on
Yeah.
here. Another one of these Brisingid sea stars. Lots of feather stars underneath. Look at the bioturbation in the rock in
Amazing, isn't it?
these turbidites.
Yeah. Wow.
Oh, Swiss cheese.
termites.
Yeah,
Termites.
termite problem.
Yeah, deep sea termites.
Wow. Kevin, can we go in there?
I'll try.
Yeah. No, we didn't lose Sebastian when the alarm went off. It was all okay, really. It was fine, wasn't it, guys?
What's that?
When the alarm went off, everything was fine.
Yeah, you know, it's good to get your heart rate.
Yeah, it woke us all up. Yeah. No, it was all good. Wow, look how steep this is. Yeah. I wonder where we are in relation to the crest. Like how far? Still a little, still a ways to go. Well, I think it was 1370.
1370 it was, yeah.
Yeah.
Anyway, we ended up covering pretty good ground.
Yeah, the dive started around about
1772.
1772, and we're looking at about 1370 to the crest of this. Here we go, and you can see, in fact, you see the inset. Excuse me, the inset. The little yellow box is us. Little ways to go. Gee, we'd have to move quick to get to the top. We were trying to see what was at the very crest here. But certainly the steeper part has been really interesting. Lots of exposed turbidites, plenty of things to grip and hang on to here.
Looks like another sponge there.
I mean, we knew it was steep in the multi-beam mapping data, but it still doesn't quite do it justice until, oh look, the whole thing has broken off.
The whole entire block.
Yeah.
There.
You land on, see if you're not gonna help.
You know I'm gonna say yeah, sure.
So we've had undescribed ctenophore today, new ctenophore.
Yeah, been quite a good collection of cephalopods. We've seen two dead nautilus shells, and we collected one. We've seen two octopus, a big red one to begin with, and then
That's
that
right.
quite
Okay.
large grey one. I mean, really big. Lots of different fish. Thanks, John Pognoski from CSIRO. He's been one of our principal investigators, been watching from ashore. It's great to have you join us. What other highlights today?
The new carnivorous sponge, nice big carnivorous sponge
Yeah.
we got. It's an undescribed.
ROV Dive.
The sea pigs?
Oh, the sea pigs, yeah.
Yeah, lots of sea pigs. Because you got to get a little close.
Yeah.
Got to get close to the seafloor to see all the sea pigs, but there's plenty of them there.
Oh, the tunicate. Thanks, Dougal.
Yeah, the weird looking tunicate. But overall, I think it was the amount of vegetation on the axis floor. For me, that was really amazing to see all of that shallow water organic material down on the seafloor, and it was being used. Right, there were animals burrowing into the wood. There were a lot of crustaceans, a lot of those white squat lobsters. Do you remember
Yes,
that?
that's right. That's
Coconuts.
Right. Yep.
We picked the coconut up. You could see animals boring into the coconut. Clearly, a lot of organic material ends up shallow. Vegetation, plant material ends up in the deep ocean. So. It would be quite interesting to try and quantify that. How much of that organic material, that carbon, ends up down in these deeper canyons?
Yeah. Okay, there's also a comment, thank you, from Unknown Explorer that the urchin could potentially be new as well.
Yeah,
Are
okay.
Is that the big one we were
Yeah.
talking about? That was about, oh gee, it was nearly 30 centimetres,
Yeah,
wasn't
it's
it?
huge. Yeah, we saw a few of them. The urchins were interesting. Yeah, but look at this rock face, or
that duplicate again?
sediment, sandstone. It's pretty soft when you touch it. But there's plenty of things clinging to the edges. So we're really pushing now, trying to get to the very crest of this sidewall, and that means it's like we're climbing to the top of a mountain. So we didn't collect any of the coconuts, but we did do some sediment push cores right into the heart of these big clusters of vegetation. So we've got good samples that we're bringing back on board this afternoon.
Little bit of concern there about our rate of ascent, worried that we're going to get the bends.
Yeah.
That's the beauty of having an ROV.
That's
We
right.
don't need to worry about that.
Oh yeah, the
Oh,
brachiopods.
the brachiopods.
Yes,
yeah.
of course.
That's
Now we have seen a few brachiopods on other expeditions,
Yep.
but not as many. Right? These were, like the whole under the overhangs were just,
that's right.
multiple dozens of these brachiopods, and I remember how excited Carsten Luther was about these. He's a German paleobiologist who came out to Australia in 2009 on a big German-funded expedition that I was involved with, and he was particularly interested in trying to find these relict marine life like these brachiopods. And today we've seen lots and lots of them.
Almost there, great.
Yep, tripod fish, that was a good look too. There's a black coral. Sorry, can't stop, Jeremy. Got to keep going. Whoa,
There we
what's
go.
this?
Look at that.
Wow,
Wow.
okay. Gee, that is. Is that a rock that's broken off and just sort of slid to a stop? Or got some nice silicious sponges over here in the middle.
Yeah.
Oh, actually, there's quite a few of them. All these white tube-like colonies. These are hexagonal sponges with feather stars hanging off them.
Marco's on.
Marco,
Yeah, Marco.
Everyone says hi, Marco.
That was his last name, right? Yeah, that's Marco.
That's pretty, really pretty.
Thanks for the fish summary, John. The good thing is that in the YouTube video, the chat is saved with it as well, so we can go back afterwards when we're looking at the video after, and we also have the chat with the comments about the fish IDs and what have you as well.
Yeah, and looking ahead, what we've got, our event logger is taking photos of all the significant animals. Plus we've got Squiddle running, which is a software program. We're getting 5-second screenshots from the 4K vision. So really high resolution still images every 5 seconds. So
Make it, do it, do it.
Squirrel.
must be getting near the top now.
Yeah, so close.
And you've got, this is the situation camera and the inset. So this is a wide angle view. So it's looking upslope so we can see what's coming ahead of us.
We're on borrowed time now, so we appreciate that. We are so close.
There it is. You can see it.
Yay.
You can see the crevice. Make sure you land on
There we go. This is the top of the mountain.
Plant a flag.
Yeah. Well, it's all part of it's all within the Great Barrier Reef Marine Park.
There we go.
So this is the crest of that canyon, And if we were to keep going, it would just drop down to
The other side.
another canyon. There's a lot of them here. They're all exactly side by side, but we've now climbed one full wall. This is a giant isopod burrow in front of us.
All right.
Anyway, look,
We should sign off.
yeah, we should. It's been a long day, and thank you very much for everyone joining us ashore. We really do appreciate all the comments and certainly the expert opinions. Plant that flag.
There we go.
Oh, look how
Sebastian was
soft
here.
that is. Yeah. You're signing your name, are you, there, Chris?
Dear, I just
JR.
There we go.
Very good.
That'll be preserved in the fossil record for millions of
Yeah,
years to
that'll
come.
be there for 1,000 years. Thanks, J-Rod.
Thanks, Jared. Thanks, everyone.
All right, we'll
sign
catch
off.
you at our next dive. Hopefully, maybe in a few days' time. Everything, of course, is weather permitting, and so thank you very much, everyone who joined us for the our third expedition to the Great Barrier Reef, called the Northern Depths of the Great Barrier Reef.
Hashtag
Hashtag
EdgeGBR.
EdgeGBR.
Thanks, everyone.
All right, bye.
good job.